What is Baeyer-Villiger oxidation? Baeyer-Villiger oxidation is a type of reaction which involves oxidation of ketones, either the aliphatic to form carboxylic acid esters or cyclic to form corresponding lactones. The common use catalysts for such type of the reaction are peroxycarbonic acids in organic synthesis or enzymes so-called Baeyer-Villiger's monooxygenases in applied biocatalysis. So, on this lecture, I'm going to speak about a biocatalytic approach of BV oxidation, where as the biocatalyst, the recombinant cyclohexanone monooxygenase was produced by Escherichia coli in a form of wall cells under non-growing conditions. The reaction scheme of the model reaction can be described as three substrates, second and third, involving two main enzymes, a coenzyme and a cofactor, where the oxidation takes place right here. The initial step of the oxidation of the ketones starts here, binding the molecule of oxygen with a reduced form of FAD, which form a reactive peroxyflavin complex. And further, this complex perform a nucleophilic attack on a carbonyl functional group, resulting in a further alkyl rearrangement where the CC bond is cleaved and the atom of oxygen is inserted. But subsequently, after this happened, lactone is eliminated as a product, but the coenzyme remains in oxidized form of FAD+. This form of coenzyme is not able to bind another molecule of oxygen as here. So that the oxidized form of coenzyme has to be reduced back, performing the reduction by NADPH. But again, because of the redox reaction, NADPH remains in an oxidized form of an NADP, which is regenerated in a glycolysis cycle of glucose. So, to sum it up, the biocatalytic BV oxidation is a nice network, enzymatic network, appearing high potential in optimization studies. The entire process, the entire system was maintained as the upstream process design and optimization in a lab scale. Well, the investigation can be divided into 5 distinct phases, where on this lecture I'm going to mention only these 2. However, all these sections have something the same, and our bacteria cells knew it all the time, but we didn't. And it is that a crucial factor is oxygen transfer rate. But the question is why? As I already mentioned, the biocatalyst we used was in the form of wall cells, the resting cells. E. coli is characterized as a facultative anaerobic microorganism, but in oxidation processes, it is essential the oxidative adjustment of the bacteria. So that it means in biocatalytic BV oxidations, the oxygen is consumed in 2 simultaneous ways. The first one as an essential important nutrient in the metabolism to keep the cells viable, and the second as the substrate of the oxidative reaction. Because of this knowledge, the oxygen uptake rate in metabolism was evaluated, adopting the initial reaction rate method. From the progress curve, the oxygen uptake rate was evaluated. Here we can see the decrease of the concentration of oxygen in time. This was conducted for a various concentration of the biocatalyst, where the specific oxygen uptake rate in metabolism was evaluated. Furthermore, the oxygen demand in metabolism is already known, so now we can talk about the role of the oxygen in the reaction, which open up the possibilities of the reaction BV oxidation reaction condition optimization. As you can see, the investigated parameters for the optimization study were concentration of the biocatalyst, initial concentration of the ketone, which was our main substrate, and the volumetric oxygen mass transfer coefficient, KLA. Several essential conclusions were made and drawn here. At first, we can see that a specific reaction rate or specific activity, respectively, demands on the oxygen transfer rate. It's quite a clear statement because, as we said, the oxygen is consumed in a metabolism and also in the reaction. Whereas we see the insufficient oxygen transfer rate results in lower specific reaction rate, respectively specific activity. So to maintain the specific activity the same, we have to choose the adequate or corresponding KLa value to maintain the activity unchanged. Moreover, it was found that the ketone concentration stands for a strong inhibition effect, where, as you can see, initial concentration of ketone exceeding 4 grams per liter exhibit inhibition, whereas the selectivity of the reaction is lower. But as you can also see, this trend is clear in respect to biocatalyst concentration, so that we conducted 2 additional experiments, certainly with an adequate or corresponding KLa value, so we have now oxygen limitation. And this trend was confirmed where we achieved nearly 93% of the selectivity. So therefore, at this point, we described or we talked about the importance of the oxygen transfer rate for the biocatalytic BV oxidation when the biocatalyst is in the form of wall cells. This knowledge enables us to design the, let's call it, model or develop the design equation for the process parameters. At first, let's talk about what we know so far from the optimization studies is that the maximal specific activity we are able to prepare as a form of biocatalyst by optimized cultivation and induction conditions is 55 units per gram, which when we want to do a dynamic model, we have to recalculate as a reaction rate, or in this case, rate of the lactone production. Secondly, the material balance is every time possible to write. Maybe it looks quite strange, but this is a material balance of oxygen. Okay, at this point, we want to design or develop the design equation. And we have 2 equations where we must choose one dependent variable and the other variables must be taken as independent. In this case, the biocatalyst concentration is independent variable because based on this value, we have to adjust the oxygen transfer rate. And why? Mainly because our biocatalyst, living wall cells. Okay, well, let's continue. So When we express OTR as a function, biocatalyst concentration, also replace the activity and production rate as a function of biocatalyst concentration, we get such form. Then let's adapt the maximum specific activity we are able to prepare the biocatalyst. And it is observed that the relationship between biocatalyst concentration and oxygen transfer rate is a linear function. However, OTR can be expressed by a process parameters as KLA, what is our design parameter, and saturation concentration of oxygen. This is a process parameter that we are able to experimentally determine or using CFD simulations, for example. And saturation concentration of any gas is a constant under isothermal isobaric conditions. So, in summary, the design, or let's call it the graphical expression of the design equation, looks like follows. As you can see, for the various specific activity of the biocatalyst, when we want to exceed the oxygen limitations, either from the metabolic way or from the catalytic way, we must, for example, when we take 10 grams per liter, choose at least 40 millimoles per liter per hour of oxygen transfer rate. Here is the summarized for our system. And this, let's call it approach, is applicable for any direct oxidation involving wall self-catalysis. Okay, whereas I was talking a lot about the oxygen transfer rate, let's focus a little bit on oxygen mass transfer phenomenon. Volumetric oxygen mass transfer coefficient is experimentally to determine very straightforward. And as long as it is an important parameter for us because it is our design parameter, we have to know its value. The KLA values were determined by adapting the gazing out method, where the oxygen concentration was measured in time, and after that, the calculated value from the rate equations were compared, and the KLA were fitted for each point. Moreover, KLA as a parameter is a product of two other variables, KL, which stands for a liquid site mass transfer coefficient, and A, which stands for a specific interface surface area, which is directly dependent on gas holdup and specific surface area of bubble. These two variables are pretty tricky because their determination on evaluating of measurement are typically challenging. But we found out the simple method using a high-speed camera footage to analyze via image analyzer each capture, and the dimensions of these bubbles, as you can see, were measured, dimensions were evaluated, and the specific surface area was calculated from the sample of 30 bubbles. There I can offer you a short footage from the high-speed camera. In my opinion, it's pretty satisfying to watch as a screensaver, for example. Okay, let's continue. Here you can see the footage or the captures for the impeller frequency range from 200 RPM to 1200 RPM for the aeration rate of 3 ppm. From the first side, what you can see is that increasing the mixing speed, the gas holdup is rising. Results confirm it, and you also can you also are able to confirm it by the free side. What does it mean? The higher the gas holdup is, it's prolonging the contact time between the gas and liquid phase, making mass transfer more effective. At the second point, higher impeller frequency causes higher turbulence and bigger shear forces, which are, I don't want to say damaging the bubbles, but they are forming a smaller bubble. Also, you can see that this is the biggest one bubble, and here are pretty small bubbles. What does it mean? The smaller the bubble is, the higher the specific surface of the bubble is. So that if we take the longest contact time expressed as a gas holdup and the smallest bubble we are able to generate, we should get the highest or the most effective oxygen mass transfer equals to the highest value of KLA obtained. Okay. To sum up the results, oxidation processes involving wall self-catalysis are strongly influenced by oxygen transfer rate, which is quite a logical fact. So that if we are designing a process involving oxidation process, and we also have a wall cell catalyst, the design parameters influencing KLA should be proportionally considered in purpose to maximize the process productivity, for example. Okay, thank you for your attention.